Thanks for the introduction, and I'd like to thank Ken for the invitation. I met Ken at a meeting in Wyoming, Jackson Hole, aimed at trying to come up with new ideas to improve cognitive performance in particularly the military and people who have to keep focused on a task for extended time periods. And one of the approaches that might help, that I'm going to talk about today, is intermittent fasting, or reducing energy intake or eliminating energy intake for extended time periods. And I'm going to talk about this from an evolutionary perspective, that is to say, why it is that it makes sense from an evolutionary perspective and what the data are that your brain should be working well when you're hungry and haven't been able to obtain food for a long period of time. If your brain isn't working well, you're not going to be able to compete with your competitors to obtain the food. And I'm going to talk mostly about work we've done in animals where we can tightly control their food intake, their eating pattern, their level of exercise, and we can directly look at their brain, look at individual nerve cells in their brains, put electrodes in those nerve cells, study communication between the nerve cells, take out the brain tissue, measure levels of various neurochemicals to try to understand why it might be that fasting and exercise are good for the brain. And then I'm going to talk a little bit about some of the translational work we've done, asking a couple simple questions in human subjects. Can human subjects maintain intermittent fasting diets long term? And if so, does it improve their health? And then hopefully, if I don't forget about it at the end, I'm going to mention a study we have ongoing where we're focusing on the brain. Okay. Here we go. Okay, evolutionary perspective. You don't see many fat predator or prey in the wild. Okay, and so if you're a hungry bobcat and you haven't been able to catch a rabbit in a long time, you better figure out how to track down the rabbit and catch it. And moreover, you better be physically fit and be able to catch it. And on the other hand, if you're the prey, likewise, you better figure out how to avoid the predator. So it just makes sense intuitively that the brain has to be functioning well and you have to be physically fit for maximum performance to survive, at least in the case of animals in the wild and probably in the case of our human ancestors before the agricultural revolution. Okay, here's another example. We have an eagle swooping down to get a fish, and so in addition to being able to recognize visually and detect the prey, they have to have a good idea where to look and what kind of patterns of rippling in the water represents a fish versus some other disturbance in the water. Then also it's important, social interactions are very important in survival among many species. We see some wolves here surrounding a buffalo. So they found that if they worked together, they could kill an animal that an individual wouldn't be able to kill by itself. So this is an example of the evolutionary basis of social interactions, but again, all revolving around figuring out how to obtain food. This is a major force for evolution of the brain of many organisms, including humans. Okay, our arboreal non-human primate ancestors. So before we became bipedal, our ancestors lived in the canopy, and they had to learn where sources of the food are. And I'm going to come back to this point in the end because it's kind of interesting from an evolutionary perspective. So these primates had to know where fruit trees were. They also had to know at what times of the year different trees, the fruits were ripened, and there's even evidence that a good ability to know when and where to find these sources of food was a driving force for evolution of what's called the prefrontal cortex, which is an area of the brain that has expanded most in our primate ancestors and in humans, and it's a brain region involved in what's called executive function and decision making, which humans do really well. In fact, there's whole books written on the evidence supporting this notion that our non-human primate ancestors, the reason their prefrontal cortex started to develop was these being able to make decisions on how to obtain food with limited resources. So then we can go even to, I'm not going to spend much time on this slide, but I could even make the case that development of tools was primarily early on devoted to making tools that enabled us to get food and compete to get food. Bow and arrow. A complex weapon for hunting. Now we have guns, and we use guns not only to obtain food, but unfortunately for more nefarious purposes. Mass collection of food, right? Mass production of food, which is good and bad. It's good and it makes it easy to get a lot of food, nobody starves. Bad, and because you have big agri and refining, making corn syrup, and people have constant access to high energy density food. They don't have to work for their food. They don't have to challenge themselves to get food either physically or mentally. And we think this is not good for the brain in the long run. It's better to challenge yourself physically and maybe by fasting, which will activate mechanisms in the brain that I'll talk about now, which seem to be good for learning and memory, and we think may reduce the risk for developing age-related neurodegenerative disorders such as Alzheimer's and Parkinson's diseases. Okay, processed foods. Okay, so this is just a quick summary of this. The point here being that in the past, animals and our non-human primate ancestors, and even hunter-gatherers, humans, had to work for their food and had to go extended time periods without food based on the seasons and other aspects, drought, weather and so on. And as you know, the major killers in humans 100 years ago were infectious diseases, actually injuries, and cardiovascular disease and diabetes were very rare back then. And now with the processed food industries and technological advances that allow us to go our whole life, earn a living without expending any energy. You go to your car, drive to work, take the elevator, sit down all day, come back, and with ready access to food. But now the major killers are cardiovascular disease, cancers, diabetes. Much of this driven by the couch potato lifestyle. I would also point out here that, and I guess I didn't put the slide in, with advances in research in the areas of cancer, cardiovascular disease, and diabetes, so for example, early detection of some cancers, colon cancer is a good example, and where you can detect it early, get rid of it before it kills you, and treatments for cancers, and then cardiovascular disease, people who would have died in their 50s 80 years ago are now getting bypass surgery, living another 20, 30 years. Same with diabetes. There's some effective treatments, although none of them are as effective as exercise and reducing energy intake. So many people who would have died when they're in their 50s and 60s are now living into their 70s and 80s, which is the danger zone for Alzheimer's and Parkinson's disease in terms of age. And unfortunately, there are zero treatments that are effective for Alzheimer's in slowing the progression. Parkinson's, there are symptomatic treatments early on in the disease, but invariably the patients will have progressive degeneration and die. So this is a huge problem, not only from the demographics of the baby boomers reaching the danger zone for these brain disorders of aging, but also from the fact that advances in allowing people who would previously have died at earlier ages to live to these ages. Okay, this is a schematic diagram showing the nerve cell circuitry of a brain region called the hippocampus. It's a brain region critical for learning and memory, and the reason is because it receives inputs from our different sensory inputs: vision, hearing, touch. All of this sensory information funnels into the hippocampus. And so since that's where these inputs converge, that's where the pairing of an image and a sound, for example, are paired temporally. So when you look at my slide and you listen to my voice, then that visual and auditory information is converging, and what happens here in the hippocampus is critical for at least the short-term memory of that, so that 20 seconds from now, I can change the slide and I can ask you verbally what was on my previous slide, and you can give me a visual description of what was on the slide, and that's because of this important brain region, the hippocampus. This is a brain region where nerve cells become dysfunctional and degenerate in Alzheimer's disease. Now, it's been shown by other labs in our lab that vigorous physical exercise, like running, for example, and dietary energy restriction, and also intellectual challenges in doing what we're doing now, increase excitability of these nerve cells in the hippocampus. And what happens is there's a neurotransmitter represented by the red dots here, I guess, called glutamate. It's an amino acid, but it's also the major excitatory neurotransmitter in the brain. So it's released from an axon terminal at a synapse, and it activates receptors on the subsequent cell in the circuit, and you get flow of information. And also with activity in these nerve cell circuits, this electrochemical activity, that stimulates production of a protein called BDNF, brain-derived neurotrophic factor. This neurotrophic factor, its levels increase in response to exercise, intermittent fasting, and intellectual challenges. And it's been shown in animal studies, and also there's some evidence, I'll briefly show you one slide from human studies, that BDNF plays a very important role in learning and memory. So if you block BDNF production or its action, that will impair cognitive performance in animals. And there's also correlations between BDNF levels and cognitive performance in humans. Another interesting aspect of this hippocampus is that there are stem cells, dividing cells that can replicate, and then when they get the right signal, such as exercise or fasting, these cells stop dividing and they form new nerve cells, which are then integrated into the nerve cell circuits. And one of the investigators in my group, Henriette von Prague, has shown that the production of these new nerve cells in response to exercise enhances a form of learning and memory called spatial pattern separation, and I'll show you one or two slides on some of those data. And this becomes very important in thinking about how exercise might benefit human cognition, even higher cognitive functions such as imagination and creativity in humans. And I should also point out, this intellectual enrichment, this can be, thinking about complex problems such as we're thinking about now, or it can be music, learning and playing music, recalling and maybe improvising in music. So that's another type of environmental input that seems to activate these pathways that enhance synaptic plasticity and promote the production and integration of new nerve cells. Okay, I'm going to briefly talk about exercise before I talk about intermittent fasting. Dr. von Prague showed that if she puts running wheels in the cages of mice, and then identifies the number of stem cells in the hippocampus. Each black dot here is a stem cell. I don't need to go into the technical details of how we label these stem cells. She finds that running increases the number of stem cells, and then when she follows over time what happens to these stem cells, she finds that many of these stem cells form new nerve cells. So this is an individual nerve cell with the cell body here, the dendrites here. These receive information coming into the hippocampus. And so this is my son here, Elliot. And this is my daughter, Emma. I used to coach high school cross country. I quit doing that a number of years ago, actually, after Emma graduated. But I would tell the kids on the team that, this is really good for your brain. And all the parents, so we have pasta parties the day before the meet, and kind of the carbo loading type thinking. I'm not sure that's necessarily right, but anyway, but all the parents, including us, we noticed during the cross-country season, the kids, their mood is better. Exercise has an antidepressant effect. It improves mood. And we think that actually occurs through increasing BDNF levels. There's good evidence for that. In fact, the most commonly prescribed antidepressants, Prozac, Paxil, et cetera, increase BDNF levels in the brain, and at least in animal models, if you block BDNF, then these antidepressants don't work. But also, their grades are better during the cross-country season. Okay, so now I'm going to go back to the study in mice. So one way we can test cognitive function in mice is using touchscreens. So this is actually very new, only in the last 5 years or so this started to become widely used. So first you can teach a mouse that if it touches its nose to a touchscreen, it will get a food reward. So these are small boxes with a touchscreen, and then just like half a foot away from the touchscreen, a food reward bin. And this is all computerized. Then what we do is we put one lit icon on the screen, and over a period of a couple weeks, the mouse will learn if it touches this icon with its nose, it gets a food reward. And then we put two icons on the screen. We train it to touch this one for a food reward. If it touches this one, it doesn't get a food reward. Then we decrease the distance between the lit icons. And as we decrease the distance, going from big to small icons, the mice make more errors. They have more trouble distinguishing which one they should touch because they're closer together. So we call that spatial pattern separation ability. And however, if we put running wheels in the cages of the animals here, they make fewer errors, significantly fewer errors, as we decrease the distance between these icons. So running improves the ability of the mice to distinguish between these icons when they're at close distance. And we found a correlation with production of new neurons. That is, fewer errors, more new neurons. Okay, I'm a trail runner, and from an evolutionary perspective, human evolution, trail running is the quintessential human experience. It's what our ancestors did a lot of, like walking or running on trails. And it became very beneficial and the data suggests that we lived in small groups, tribes, hunter-gatherer type scenarios. And so it was a big advantage for us when we're running, going some distance, say 20 miles away from the where the tribe lived, to be able to remember what we encountered when we went on that trail run or walk. So that and I think this was actually a driving force for development of language, or at least one driving force, because it's obviously of value to transmit the information on what you saw and encountered to your tribe mates, so that then they can go out and we can tell them to you go on the blue trail, you go on, let's see, where do we go? So our running group, I have a running group Saturday mornings. We go trail running. So for example, we'll say, okay, we're going to take the yellow trail to the blue trail, and then we're going to come back, or we're going to go this route or the other. And after a few runs on the trail, not only do you remember the general route, you remember details of where individual rocks and roots are, and what's coming up. You know, there's a big hill coming up, so maybe I should back off now because I don't want to be dead at the end of that. But from the standpoint of cognitive function, if you have to be focusing on what you're doing while you're exercising, I think that promotes optimal brain function in the long run. No pun intended. Okay, so let's go back to animal studies. Diabetes is a big problem now, and no pun intended again, but you know, being overweight's a big risk factor for diabetes. So we have animal models of diabetes. One model is leptin receptor mutant mice. Leptin is a hormone. When you eat a meal, it's released from fat cells and into your blood, and it travels to your brain, and it tells you to stop eating. You're getting full. So obese people often have what's called leptin resistance, and they actually have high levels of leptin. So they produce the leptin, but the leptin is not effective in telling the brain to stop eating. So they continue to have the perception that they're hungry, even though they're clearly not in danger of starving. But they, you know, so this is and there's also another problem with diabetes, which is well understood. It's called insulin resistance. So individuals with type 2 diabetes often have elevated insulin levels, and the reason is because, for example, the muscle and liver cells that normally respond to insulin by taking up glucose from the blood and moving it into the muscle or storing it in the liver, those cells don't respond to insulin properly, and so your pancreas starts putting out more insulin to try to get the glucose out of the blood. Okay, so anyway, we have mice that can't respond to leptin, and so they have constant hunger, essentially. And they overeat, they become obese and diabetic. And so what we did is we looked at these neurons in the hippocampus, and we looked at individual dendrites, these regions that receive information coming into the hippocampus. And so this is one dendrite here, and along this dendrite you'll see these perpendicular projections. And each of these perpendicular projections, which are called spines, represent one synapse. And then we simply count the numbers of these synapses, and we quantify this. And so in this experiment, what we had is 4 groups of diabetic mice and 4 groups of normal mice. We call them wild-type mice. One condition was the normal laboratory housing conditions. Ad libitum fed. They can eat whenever they want, eat as much as they want. And sedentary, no these small cages, no running wheel. Then we had a group, they were fed ad libitum, but had running wheels in the cages. We had another group, we reduced their daily calorie intake by about 30%, but they were sedentary. And then finally we had a group that was calorie restricted and they had running wheels in their cages. And if we look at the normal mice here, you see that the black bars, calorie restricted group had increased number of synapses. The animals with running wheels in their cages had increased number of synapses compared to the couch potato mice. And then the combination of calorie restriction and running had the maximal effect in increasing the number of synapses. And in fact, these animals do the best in learning and memory tasks. Then we look at the diabetic mice here, and you see overall they have much lower number of synapses than do the normal mice. However, nevertheless, both calorie restriction and running increased, approximately doubled the number of synapses. And this isn't immediately, this occurred over a 3 to 4 month time period. And then finally, in this lower panel, we see a nice correlation between synapses and BDNF levels. So calorie restriction and running increase BDNF levels. The combination, you get a further boost in BDNF levels, and we think BDNF is playing a role in the improved learning and memory. And what about humans? Well, it turns out there are certain families that have reduced levels of BDNF levels because they have a mutation that they in the gene encoding BDNF that they inherited from one of their parents. So these are BDNF heterozygous humans. And then if you compare, and this study was done in young adults, so high school kids, either normal kids or kids with reduced BDNF levels, and this is testing their cognitive functioning. So there was a statistically significant poor performance of the kids with reduced BDNF levels compared to the normal kids. And then this study was done recently, published in 2013. Autism is a very big issue and problem now. So they also tested these kids for behaviors that are characteristic of autism, and the kids with reduced BDNF levels had significantly higher score on these autism-like behaviors compared to kids with normal BDNF levels. So these data we're obtaining in mice is not just relevant to mice, we think it's very relevant to humans. Okay, I mentioned in humans that we have a big prefrontal cortex. Also, two or other regions of the brain that have expanded in humans are those that are related to language, either indirectly or more directly. So visual cortex and associated regions. So language is essentially processing of visual and auditory information and correlating them. You read something, and it's all recognizing patterns, the letters, and the sequences of patterns. Language, speech is also the same thing. In that case, sequences of sounds. Okay, so I wrote this kind of provocative article. If any of you are interested, you can access it easily on the internet. It's called Superior Pattern Processing is the Essence of the Evolved Human Brain. So I showed you that exercise and also intermittent fasting can improve cognitive function and spatial pattern separation. And I think even things like creativity, imagination, being able to foresee events scenarios that might happen in the future, and decision making all revolve around processing visual and auditory patterns. Okay, now I'm going to talk about fasting. And so I gave a TED talk on this, so if you're interested, you can find this on the internet easily. But from a historical perspective, there's many cases where you can find famous people who claim that they did their best thinking and work when they were fasting. So Plato, for example, he found that he did his best thinking and writing when he was fasting, so he subjected himself to sort of intermittent fasting type scenarios. Gandhi, and also many religions, fasting is commonly practiced, and because it clears the mind. We think that's actually true, and from an evolutionary perspective, a non-religious, true to evidence perspective, fasting should be good for the brain from the standpoint of being able to compete to obtain food and resources and outcompete your competitors. But also, a lot of interesting quotes. For example, Mark Twain, little starvation can really do more for the average sick man than the best medicines and best doctors. And so on. Also, very interesting, Upton Sinclair, who wrote the book The Jungle, that exposed problems in the meatpacking industry around the turn of the century, 1900. He also wrote a book called The Fasting Cure and published it in 1911. Very interesting. He interviewed 200 and some people who had some ailment. They went on some fasting diet of various types and almost uniformly found benefits for their health. You can find the full text of that on the internet too. Okay, in the 1990s, we were working on trying to understand what's going wrong in the brain in Alzheimer's disease, Parkinson's disease, mainly in animal models. And we either used models that are based on putting a mutated human gene that causes Alzheimer's disease, for example, a mutation in the amyloid precursor protein gene, or a mutated gene that causes inherited Parkinson's disease, for example, mutation in a gene encoding the protein alpha-synuclein. We put those genes into the genome of mice, and those mice, in the Alzheimer's model, they'll get accumulation of amyloid in their brains as they get older, and they'll have cognitive dysfunction. The Parkinson's model, the mice will develop accumulation of this alpha-synuclein protein in their dopaminergic neurons and other neurons, and they'll develop motor dysfunction and actually die. And then we also had neurotoxin-based models where we administer a toxin to the animals that selectively damages nerve cells in the brain regions in which the nerve cells degenerate in the human disease. So, for example, in this, one of the first papers we published on intermittent fasting, we used toxin-based models of severe epileptic seizures and kind of an acute amnesia, and then a model of Huntington's disease that's based on using a toxin that impairs the mitochondrial function in nerve cells. So what we do in these experiments is very simple in design. We have two groups of rats or mice. One group, they eat every day as much as they want. The other group, we have them fast every other day. So one day we go in, we remove the food from the cage. The next day we go in at the same time, we put food back. Next day remove it, next day put it back. So they go 24 hours no food, 24 hours food. That's the way they go for months and months. And then we expose them to a toxin, or we follow the outcome as the disease develops in the genetic models. You know, we test their learning and memory, we test their motor function, and so on. And so, for example, in this study, we found that this intermittent fasting, alternate day fasting, is neuroprotective and improves functional outcome in models of severe epileptic seizures and Huntington's disease. This is a different model of Huntington's disease where we have mice that express the mutated human Huntington gene. This is a model of Alzheimer's disease here that we developed about 13 years ago. These mice develop amyloid accumulation and cognitive dysfunction. Interestingly, we found that the intermittent fasting prevented the cognitive impairment as these animals got older, but it didn't prevent the accumulation of the amyloid. So somehow, and we think we know in part how, the intermittent fasting is protecting the neurons against the amyloid. And that's very interesting because it turns out that there are many humans who, they live into their 80s, 90s, even 100. They're sharp as a tack, but then when you look at the brains at autopsy, they're loaded with amyloid. So somehow those individuals resist their nerve cells are resistant to the adverse effects of the amyloid, whereas in other individuals, the nerve cells are not resistant. And we think in the case of exercise and fasting, we have some insight into why, and I'm going to show you in the next slides some of that. And we also found, for example, if animals that have been maintained on intermittent fasting diet, they have a better outcome following experimental stroke. There's less brain damage when we block off the blood supply. Fewer neurons die in the territory supplied by that, mainly by that blood vessel, and they have improved functional outcome. Okay, so what might be the mechanisms whereby fasting is good for the brain? Well, we know one major thing that happens from the standpoint of energy metabolism is that if you eat 3 meals a day plus snacks, every time you eat, the energy goes into your liver. It's stored in the form of glycogen, and that's always tapped into first. The glycogen is metabolized and released, the glucose is released into the blood. So if you eat 3 meals day plus snacks, you never deplete the glycogen stores in your liver. However, if you go, for example, say 16 hours without eating, certainly by 24, you've completely depleted the glycogen stores in your liver. Then what happens is fats are mobilized. So fatty acids are released from your fat cells. They go into the liver, and they're metabolized to what are called ketone bodies, such as beta-hydroxybutyrate and acetoacetate. And so then, and you can, we all know this, you can survive for weeks, and many people a month, not eating anything, as long as you have water available and don't become dehydrated. And the reason is that you're running, if you will, on ketones. Okay? And it turns out ketones are an energy source for neurons similar to glucose in that both glucose and ketones can be converted in the nerve cells to ATP, adenosine triphosphate. And ATP is what the molecular energy substrate is that allows our muscle cells, our brain cells to work. Okay, ketones are good for brain cells, not only because they're an energy source, but we think they actually have signaling functions. It's a signal that's coming from the blood to the brain that's alerting you, and in the case of our animal studies, the animals, to the fact that there's this shift in energy metabolism, which is an indicator that you better start finding a new food source because eventually you're going to run out of fat stores too, and then you're in big trouble. And we find, in fact, that ketones increase BDNF production in the brain, and we think that ketones are one peripheral signal that alerts the brain that it better start working well. Okay, so we did all these studies in animals, and then I was, through correspondence, communicated with a number of MDs who work mostly with overweight subjects. For example, Jim Johnson worked with asthma patients. So we did a study with him where we took a small study, 12 asthma patients, they're overweight. We put them on a diet where every other day they only eat 500 calories, which is kind of a very severe change. These are overweight subjects who are normally eating 3 meals every day, and they're taking in too much energy. But 10 of the 12 subjects were able to go 2 months on that diet. They lost weight, as you can imagine. Their asthma symptoms improved. Their airway resistance improved. Jim Johnson sent us blood within about 2 weeks, and then particularly by a month, markers of oxidative stress and inflammation in their blood were way down and stayed down through 2 months. And so Jim wrote a book called The Alternate Day Diet based on that and published it. I just let these people publish the books because 2 reasons. They did a good job. Another reason, I'm working for you. You pay my salary. I can't make any profit on publishing a book. I'm a U.S. government employee. And then we did a study in collaboration with Dr. Michelle Harvey at the University of Manchester in England, who works with women at risk for breast cancer because of their family history, and they're also, most of them are overweight. And so in that study, there was about 100 women. We divided them into 2 diet groups. One group, they reduced their daily calorie intake by 25%. The other group, we had them 2 days a week, and in this study it was 2 consecutive days, although we don't think that necessarily have to be consecutive. But on those 2 days, they ate only 500 calories. And that's now called the 5-2 diet, or the fast diet. And so what we found, and they stayed on these diets for 6 months, and we had good compliance. Not only were they losing weights, but we measured leptin levels, we measured ketone levels, and we took blood on the days they're supposed to be eating only 500 calories, the other days, so we can tell from a scientific standpoint when they're following the diet. Both energy restriction groups lost weight, but the women on the 5-2 diet lost more belly fat, and they had greater improvement in insulin sensitivity. And very likely the reason is because on those 2 days, they're burning fat. Whereas the women who are just eating 3 meals and just reducing by 25% calories each meal, every time they eat, they replenish the glycogen stores and they never shift to burning fat. So that led to the BBC doing a documentary on this, and this Michael Mosley, the producer for the BBC, he wrote this Fast Diet book, and there's been some copycat books, and Michelle Harvey wrote a book. And then the other diet that's become popularized is what's called time-restricted feeding. So instead of focusing on maybe 2 days a week, cut way back, or eat no food but maintain hydration, another approach is every day limit the time period you eat to, say, 8 hours, 6 or 8 hours. And it turns out most Americans actually, they put something caloric in their mouth when they get up, and then they'll eat several meals, and then they'll eat a snack in the evening. So the only time they're not putting something caloric in their mouth and therefore not replenishing their glycogen stores in their liver is when they're sleeping. So the idea with this time-restricted feeding or 8-hour diet is you limit your time window that you consume food to, say, 8 hours or so. I personally kind of limit to about 6 hours. And then I actually exercise after having not eaten for 16 or 18 hours. I had to eat lunch today, but anyway, I didn't eat breakfast. I ate a modest lunch. Ken will attest to that, not many calories, and I'm feeling faint. No. No, but I'm, so you can run on ketones for quite a while. Okay, but this is the point here is that from the standpoint of challenging yourself bioenergetically from a short-term fasting approach, there are a number of different ways that people might think about incorporating this type of approach into their lifestyle based on their work schedule and so on. For me, it's easy to get up, go right to the lab, start working. I drink a lot of tea, which seems, and then I don't eat lunch. Then I work out mid-afternoon, and I find I'm fine. I don't get hungry that much once you get adapted to it, and I don't have any postprandial sleepiness, that you might get if you eat lunch, particularly if you eat carbs for lunch. Okay, I'm getting kind of long-winded, and I don't know how much time I have left. Oh my gosh. Okay. Ketones may be good for your brain. Okay, so here's kind of the idea. Now, what we're finding, and I'm not going to have time to show you any data, but if we look at the nerve cells themselves, what's happening in the nerve cells with exercise, with energy restriction, particularly fasting, and I'm going to talk about this at the end, hormetic phytochemicals. We think that mitochondria function better, and actually we think there are more mitochondria in your nerve cells if you exercise regularly. We find that exercise in BDNF will increase a process called mitochondrial biogenesis. It's the growth and division of mitochondria. So similar to what happens in your muscle cells when you exercise regularly, you increase the number of mitochondria in your muscle cells. That makes sense. Then your muscle cells they have more energy, ATP. We think that's happening in nerve cells with these types of bioenergetic challenges. Also, very importantly, we think that these challenges are stimulating a process called autophagy. Autophagy means self-eating, but essentially what happens is the cells enhance their ability to remove damaged molecules, dysfunctional mitochondria. So they're clearing out the garbage. Okay, and then neurotrophic factors increase. We've found that DNA repair ability increases. And altogether, we think that may counteract the accumulation of these proteins, amyloid beta peptide, tau, and so on, that accumulate in Alzheimer's and Parkinson's. I'm going to skip all this. Essentially, recently we showed through a number of approaches that that if we block the ability of nerve cells to increase the number of mitochondria, that will block the ability of exercise and BDNF and we think fasting to increase the number of synapses. So again, we're looking at these synapses here, and we find that if we block the ability of neurons to produce more mitochondria, we block or decrease the number of synapses. That's mainly what that is. I'm going to skip this, except to say that we discovered that there's a protein in mitochondria called sirtuin-3, and exercise and BDNF will increase the production of this protein in the mitochondria, and this protein, SIRT3, makes the mitochondria more resistant to stress, more resistant to oxidative stress, that is free radicals, more resistant to bioenergetic stress. Okay, so in the last 2 minutes, I wanted to get back to this because this is very interesting. So I haven't mentioned a word yet, a word that we've had a lot of interest in is called hormesis. And the idea here is that, and this is well documented, exposure of cells or organisms to a mild stress can enhance the ability of the organism and the cells in that organism to more severe stress. So the idea here is that low levels of stress are good, high levels of stress can be bad. And that's even true with exercise. You can overdo it in exercising. You can overdo it in fasting. You can starve to death. That would be overdoing it there. But there's a big therapeutic window for fasting. It's pretty hard to overdo it there. But from an evolutionary perspective, there's a lot of interest in what is it about fruits and vegetables that's good for health. And for many decades, the dogma is that there are chemicals in the fruits and vegetables that scavenge free radicals, antioxidants. So you go to the health food store or the vitamin, you'll see antioxidant, antioxidant, antioxidant. That's incorrect. The chemicals, at least some of the chemicals that are in fruits and vegetables that are good for your health, are good for your health from an evolutionary perspective because they are toxins. They are noxious. The reason that they're produced by the plants is to prevent you from eating the plants. And that is why the healthiest plants are the worst tasting plants, and why your kids don't want to eat the broccoli and the other bitter tasting chemicals. And so I wrote an article for Scientific American that was published last year in the July issue. And so here's the points here. I'll just read this quickly. Plants don't have the option of fleeing predators. As a consequence, they developed an elaborate set of chemical defenses to ward off insects and other creatures that want to make them into a meal. These toxins that plants produce are consumed by us. There's no question about that. And exposure to these chemicals causes a mild stress reaction, and this enhances the ability of our cells to resist more stress. So it was to our advantage to be able to eat a variety of fruits and vegetables that have even those that have a bitter taste, because they have nutrient value. They have calories, they have other nutrients. And so we develop many defenses against these chemicals. One is our tongue. They have very bitter taste. And indeed, there are many plants that have toxins that can kill us even in low doses. Okay? And we can overdose on some of the chemicals that probably everyone in this room commonly consume. Caffeine is a toxin. You can kill yourself, overdose with caffeine. If you take pure caffeine, put it on your tongue, you don't want to have any part of it. It's very bitter tasting. Okay. The second defense after our tongue is vomiting. Okay. The third defense, which is very interesting, is enzymes in the liver called cytochrome P450s that rapidly metabolize or degrade these noxious chemicals that are in the plants, the fruits and vegetables. So one pass through the liver, most of these chemicals are removed from the body. So they pass through our system once, and many of them get into the brain. So our cells are exposed to them, they induce a mild stress in our cells, and they're eliminated. So it's a transient stress. So we get this idea of hormesis. Transient or low-level exposure to these toxins is good, and high doses can kill us, but we have many defenses that prevent that from happening. So the final slide of my talk, so I talked about these challenges, what happens, fast bioenergetic challenges, intellectual challenges, phytochemicals, activate these pathways, clear out the garbage, increase neurotrophic factors, but recovery period is very important. So after fasting, eating is important because you have to get the nutrients in the cells and protein synthesis to allow growth of the cells. Building of muscles, if you're a bodybuilder, you can build muscle with intermittent fasting, but you've obviously got to eat. But as long as you eat after fasting, it's good. In fact, might even be better for bodybuilding than if you didn't do a short-term fast before you work out. After exercising, resting, sleeping is very important for the brain and particularly for the brain. And we think what's happening during these recovery periods is that's when protein synthesis goes up. That's actually when the number of mitochondria is increased. The stimulation, the pathways activated for the increase occurs with the challenge, but it's then during the recovery period that you actually get the growth, formation of new synapses, and then we think enhanced learning and memory. So these cycles of a challenge to your system and then a recovery period. Challenge, recovery, and I'm talking about within the period of a day or over a period of several days. Challenge, recovery, challenge, recovery. The best example of exercise, which is very evident, but we think it's also true with food restriction and fasting and intellectual challenges, right? Sleep is very important for, we think, consolidating what you've learned during your waking periods into long-term memories. So this is my laboratory, actually a couple years ago. So these faces are many of the people who did a lot of the original research I talked about. And this is, since I quit coaching cross-country now, NIH has an annual relay race in Bethesda. We're in Baltimore, so we never entered this race until I quit coaching cross-country. Then I started coaching, getting together teams at NIA, so this is one of the teams, and there's typically 90 to 100 teams in the race, and each leg, there's 5 runners in the race, 2 have to be female, each leg is a little over 800 meters, and so we finished first. The first year we entered, the last 2 years we finished second. The point there is we practice what we preach. Thank you very much.
Not on. Okay. Thank you so much. Now we have time for some questions and answers. Any questions out there? I will and wait for the microphone, and I'm bringing it to you.
Thank you so much for your talk. Have you looked into, or are there other parts of the research that are looking at what the effect on mental illnesses would be?
That's an excellent question. This hasn't been looked at much yet. So you're talking about fasting in particular? Yeah. Well, there's good evidence that exercise is good for many psychiatric disorders, certainly for depression, for anxiety disorders. Bipolar disorder is, mechanistically, there's quite a bit of overlap with anxiety and depression. As I mentioned, exercise increases BDNF levels in the brain, and that has an antidepressant effect. Intermittent fasting in animals, from a neurochemical standpoint, there's a lot of overlap of what's happening in the brain with intermittent fasting and exercise. So there's some scientific rationale to suggest that it's worth pursuing controlled studies in human patient populations to my knowledge, there have not been such studies done. I think it's a very good question, very important to do. There's a question that comes up, what about anorexia nervosa, which is a psychiatric disorder? And so one question is, well, this particularly affects most heavily young girls and women. And so the answer is we don't know. And I think that would be a good study, actually, have some subjects with anorexia nervosa and divide them into, try to change their eating pattern. And maybe they would be able to actually, maybe it's counterintuitive, but maybe they should actually increase their overall energy take during the week if they do some intermittent approach. But one interesting thing there that's very interesting, well, from an evolutionary perspective, obviously anorexia nervosa is not something that would be selected for in evolution. Okay, and I don't know if we go back, historically, and I guess there's no way we can know. I don't know how you could possibly know. Did anorexia nervosa exist? When did it first appear? Because obviously it's going to be selected against. And these women, of course, they stop cycling. So they're not going to reproduce. We did a study, and I don't want to. I want to give more people a chance for questions, but we can, since we're not having dinner tonight, we can.
I have my marching orders.
We can extend the question and answer period for an hour.
I don't think so.
I'm up for it.
I have to vote no on that, but go ahead.
But I'll, as quickly as I can, we did a study in rats. We had male and female rats, and we put them on either their normal diet, ad libitum. We had groups with 20% daily reduction in calorie, 40% daily reduction in calorie, alternate day fasting, and we even had a McDonald's type diet. And then we let them go for 6 months with the severe calorie restriction, 40% daily reduction in calories. The female mice, or rats, female rats became essentially emaciated. I mean, they had zero observable body fat when we ended up killing them at 6 months. The males, and they also became much more physically active in their cage. The males, on the other hand, they maintained lean mass. They even had some fat, and they didn't have an increase in activity. Now, from an evolutionary, and the females quit cycling. Their estrous cycle completely stopped. The males remained fertile. So, from an evolutionary perspective, maybe that makes sense. If you're a female and there's not much food available, you don't want to get pregnant because there's no energy for your offspring. On the other hand, if you're a male, you want to remain fertile so you can pass your genes on before you starve to death. Yeah. So, okay.
Thank you, Dr. Mattson, for your research and your lecture here. If I understood you correctly, you were talking, or you feel that there's evidence to indicate that it is increased levels of ketones such as beta-hydroxybutyrate and acetylacetate that cause the increase of the BDNF levels. If that is the case, what are the implications for a ketogenic diet and or exogenous ketones?
Yeah, so that's an excellent question. And this is a question we're trying to address in a very clear-cut manner experimentally, which it turns out not to be easy. Because I think I mentioned early on that nerve cell network activity in the brain is also increasing during exercise and with fasting. In fact, in the animals, it's very obvious. After they've fasted, the day you go in and they're very active, and you can tell they're hungry and searching for food. And we know that activity in neural circuits increases BDNF production. We also know, however, that hydroxybutyrate, even in cultured nerve cells, we add hydroxybutyrate, BDNF levels go up. So we know that hydroxybutyrate can, and we know it gets in the brain easy, we know it can directly act on neurons. So we don't know what the relative contributions are of the ketones versus the intrinsic changes going on in the brain. Now, the way to experimentally address that, which we're trying to do now, is to selectively block the production of ketones in the liver by using genetic tools to knock out the gene that's critical for conversion of the fatty acids to the ketones in the liver. So then we can fast animals or subject animals to exercise and not get the increase in ketones, and then look at the brain. And that, that's the only way we can think of the way we can really critically address that. But as far as not fast, as far as eating 3 meals a day, not exercising, but supplementing with ketones, I think there will be some benefit, but it won't be as great as exercise and fasting, because exercise and fasting have much more complex integrated effects than any single molecule can have.
All right, we have time for one last question. I'm starving, so.
We'll have 2 more questions because
Oh,
I
okay.
have great concern
Wait,
for
who's
your.
in
I want
control
your brain
here?
to function well.
Oh, that's good.
See, I'm looking out for your own interest. Okay. Okay.
I just gave the microphone to the
Oh, okay. Okay.
Excuse me, is the production of BDNF and mitochondrial replication, was there more in aerobic or anaerobic exercise, or was there no difference?
Yeah, that's an excellent question. We don't have a clear answer to that. We know BDNF does increase with aerobic exercise. In animals, it's hard to study like weightlifting in mice or rats. And the other aspect of that, in humans, whereas in animals we can take out the brain tissue and directly measure BDNF, in humans we can measure BDNF in the cerebrospinal fluid and the blood, but in the blood we're not sure where the BDNF is coming from, because it's not produced just in the brain, it's produced other places. And so in humans we don't know. And rodents we actually don't know either. So we started a study now where we have subjects, this is an exercise, but we're doing intermittent fasting in individuals between the ages of 55 and 70 who are obese and have insulin resistance. We're going to have them in 2 groups, either intermittent fasting, 5-2 diet, or normal diet. We're doing cognitive testing, looking at functional network activity by functional MRI, and we're also taking cerebrospinal fluid at baseline and after 2 months on the diets, and we can measure BDNF in the cerebrospinal fluid, and that will give us an answer from a fasting standpoint.
Great, let's thank our speaker one more time.